Hello viewers, in this video let us learn about glycolysis from the steps 7, 8 and 9. In the previous videos we have learned about glycolysis from the steps 1 to 6. Revising about the steps 1 to 6, glucose by enzyme glucokinase or hexokinase gives you glucose 6-phosphate, and this step is irreversible and regulating step. Glucose 6-phosphate by isomerization gives you fructose 6-phosphate, and this step is reversible. Fructose 6-phosphate gives you fructose 1,6-bisphosphate by the enzyme phosphofructokinase, and this step is also irreversible and regulating step. This fructose 1,6-bisphosphate is catalyzed by the enzyme aldolase to give you glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This dihydroxyacetone phosphate is interconvertible, gives you glyceraldehyde 3-phosphate by phosphotriose isomerase. So totally 2 molecules of glyceraldehyde 3-phosphate is generated per molecule of glucose. The fifth step is glyceraldehyde 3-phosphate along with one inorganic phosphate gives you 1,3-bisphosphoglycerate. And this 1,3-bisphosphoglycerate with a molecule of ADP gives you 3-phosphoglycerate plus ATP. This step is substrate level phosphorylation because you generate one molecule of ATP without entering the electron transport chain. Now about the steps 7, 8 and 9. So the 7th step is this 3-phosphoglycerate by the enzyme phosphoglycerate mutase gives you 2-phosphoglycerate. This third position, it has a phosphate group, it is shifted to second position, so it is 2-phosphoglycerate. The enzyme is phosphoglycerate mutase in the presence of magnesium ions, and this reaction is reversible. The enzyme is phosphoglycerate mutase. 8th step is 2-phosphoglycerate by the enzyme enolase by removing a molecule of water gives you phosphoenol pyruvate. It is a reversible reaction. The enzyme is enolase. Phosphoenol pyruvate is a high energy phosphate molecule. This is the 8th step. So here the fluoride is an inhibitor of glycolytic pathway because enolase can be inhibited by fluoride. Thus fluoride can inhibit entire glycolytic pathway. So fluoride is added to the blood sample collected for sugar estimation to prevent glycolytic pathway. Otherwise glucose in the blood sample will be metabolized by the blood cell and gives you less blood sugar value. So this is basis why we are adding fluoride, which inhibit the enzyme enolase. And the 9th step is phosphoenol pyruvate with a molecule of adenosine diphosphate in the presence of magnesium ions. By the enzyme pyruvate kinase, you generate a molecule of pyruvate. It is a 3-carbon compound and adenosine triphosphate. So this is also substrate level phosphorylation because you generate ATP in the steps also. So the enzyme is phosphoenol pyruvate kinase. It is a regulating enzyme of glycolytic pathway. It is irreversible reactions. So in these steps, high energy of substrate is utilized for the synthesis of ATP. So it is a substrate level phosphorylation step. So that's all about today's video. In the next video, we will learn about how lactate is generated in anaerobic pathway as well as about the fate of pyruvate. Thank you.